All right, good afternoon, ladies and gentlemen. We're going to talk about enzymes today, and we're going to try to tie in enzymes to the liver lab that hopefully you just completed. In order to understand enzymes, we're going to need to talk about what chemical reactions are, and all of this is in part of Chapter 6, Section 2 in your textbook. I'm going to talk fast because I've only got 10 minutes for the screencast. So the first question is, what is a chemical reaction? And I'll tie this to enzymes in just a moment. So a chemical reaction occurs when substances make or break chemical bonds. When we write it out on an equation form, we put the reactants on the left. A reactant is something that is reacting. And then we put the products, what it is producing, on the right-hand side. So the NaCl are the reactants, the Na and the Cl, those are the reactants. And then it forms a molecule of salt, that's the product. This particular reaction is called a redox reaction. When one atom loses an electron and one atom gains an electron, it's called oxidation reduction. Why do we mention that here? Because the whole purpose of enzymes is to speed reactions. And this is one example of a reaction that an enzyme might be able to speed up. All right, so what are enzymes exactly? Well, enzymes are protein catalysts. They're what? What's a catalyst? Well, hopefully from junior high, you remember what a catalyst is. A catalyst is anything that speeds chemical reactions without being changed itself. That means it can be used repeatedly, over and over and over again. It can speed these reactions. Enzymes are a category of catalysts, and they're made by cells, and they're made of proteins. So they produce reactions in the body millions of times faster, or they speed reactions millions of times faster than they would be under normal circumstances. In the liver lab, you had a catalyst that was not an enzyme, and that was when you used the manganese dioxide, that black powder. That was a catalyst. It sped up the breakdown of the hydrogen peroxide and caused it to bubble really fast. The enzyme that we used in the liver lab was an enzyme that's called catalase. ASE is the ending. ASE is the ending of enzymes usually, and that was the name of the enzyme that was in the cow liver. Interestingly enough, you also have that enzyme in your own body, and pretty much any animal on the planet has that enzyme. When we produce energy, the energy of our cells is called ATP. When we make that energy, we accidentally produce hydrogen peroxide. Hydrogen peroxide is poisonous to our cells, so we conveniently have this enzyme called catalase that breaks down hydrogen peroxide as soon as our bodies make it. It's kind of like a nuclear power reactor making nuclear waste, and you have to get rid of that nuclear waste. Well, hydrogen peroxide is our nuclear waste, and our enzymes break it down. We break it from hydrogen peroxide into water and bubbles of oxygen. Pretty cool how you can change something that's so bad for us into something that's actually pretty good for us. All right, so the question is, how do enzymes work exactly? We have this idea that enzymes work through something that's called the induced fit model, and I'm going to emphasize this over and over again. The shape of an enzyme is absolutely key to whether it's able to function and how efficiently it functions. So if you take a look at the diagram that I got off the internet here, that large gray piece is the enzyme itself. Let's pretend for our liver lab's sake that that's the catalase. Then the green curve and triangle shape, that's the substrate. That's the molecule that it's going to work on. In our example, it's hydrogen peroxide. Notice that the hydrogen peroxide and the catalase fit together like little puzzle pieces, and then the catalase breaks apart the hydrogen peroxide into water and oxygen. So the induced fit model says they bend and they fit together like little puzzle pieces, and then they can make or break bonds. So how is it that enzymes are so effective? What is it that they're actually doing? You need to understand this graph. There's going to be a graph like this on the test. So how do enzymes work? They lower the activation energy. That's the amount of energy needed for the reaction to occur. So let's use our hydrogen peroxide example. When does hydrogen peroxide bubble under normal circumstances? Well, if we add energy to it. I could get hydrogen peroxide to bubble if I shook it, added energy that way. I could get hydrogen peroxide to bubble if I heated it up. I could get hydrogen peroxide to bubble if I set it out under ultraviolet radiation out in the sun or something. I could get it to bubble, but that requires the red hill on the graph, that high hill. I got to add a lot of energy to get that reaction to continue to its completion. But in the presence of an enzyme, I don't have to add any energy. I don't have to heat the body up. I don't have to shake the body. Hydrogen peroxide gets broken up automatically. The the activation energy is so low when enzymes are present. So we don't need as much energy if we have enzymes present, and that allows our reactions to occur at a much greater rate. All right, so what are things I emphasized that shape is so important to enzymes. So what are things that could actually affect the shape of an enzyme? Why might an enzyme not be effective? Well, the first one you might not think of immediately, mutations. Remember that DNA tells our bodies how to build proteins. Enzymes are made of proteins. So if you have a mutation, or a person has a mutation in their DNA, it might cause their enzymes to be misshapen. The amino acids are in the wrong order. That kind of thing could happen in, say, somebody who's lactose intolerant. They have a mutation that causes their enzyme lactase, that breaks up lactose into two pieces. That enzyme might not be functioning correctly because it might be misshapen, and that could be due to a mutation in their DNA. Other things that could cause enzymes to be misshapen, heat is a big one. We boiled the liver in the liver lab and it didn't function at all. If you look at the diagram, that's because proteins melt. And I put melt in quotation marks. It's not like melting an ice cream cone, but we call it denaturing. And once a protein melts or an enzyme melts, there's nothing we can do to get that shape back again. So heat causes enzymes to denature. Obviously, we're never going to boil anything, any human tissue or anything. So at what temperature do enzymes start to denature? Well, it turns out they denature at 108 degrees, right around 108 degrees. So when somebody has a fever that's pushing 108 degrees, or they go over 108 degrees, their proteins start to denature. That interferes with their enzymes and how efficiently they work, and they can start experiencing total organ system failure if they stay at that temperature of 108 degrees for longer. The longer they stay at that temperature, the more and more of their proteins that begin to denature. The other thing that it can affect the shape of an enzyme, one other thing, not the only other thing, are changes in pH. If we add acids or bases to enzymes, we can cause them to not work well. If you remember in the liver lab, you added hydrochloric acid and you added sodium hydroxide, and the enzyme did not really work very well. When we added distilled water, which doesn't affect the pH, the enzyme still functioned perfectly. The pH of our blood is typically around neutral, around 7. And so the enzyme that we were working with, this catalase, that's the pH that it prefers, and it works best. But when you made the pH lower by adding an acid, it changed the shape of the enzyme that was in that liver, and it didn't function as well. Vice versa, when you raised the pH, it changed the shape of the enzyme, and it didn't function as well. A couple other graphs just to finish it off that are the same idea that I've just been talking about. Enzymes tend to work best at normal body temperatures. So a high fever can cause, around 108 degrees, can cause the enzymes to denature or melt. It changes their shape, and then they don't function correctly anymore. Frozen enzymes, however, this is a little bit of trivia, they don't change their shape. Frozen enzymes are okay. They just need to be reheated. They move incredibly slowly, so they don't function well. But if you reheat them, they can function just as well as they did originally. It's heated enzymes that can't work correctly. And then this last thing just talks about pH again. Enzymes work best at certain pH levels depending on the enzyme. So the enzymes that live in our blood work best at a neutral pH of 7. Enzymes that live in our stomach prefer the acidic pHs, and enzymes that live in our intestines prefer basic pHs, so high pHs. I think that's it. Please let me know if you have any questions. All right.